Hello, good morning or good afternoon, depending on where you are. And thank you for being here at the VICC Virtual Molecular Tumor Board session today. Today we have Dr. Soulafa Mamlouk from she's a group leader in clinical cancer evolution in the Department of Pediatric Surgery at Charite in Berlin. She's going to talk to us today about the use of organoids in functional studies and the evolution of cancer progression. So really excited to have you here, and thank you so much for sharing your expertise with us.
Thanks, Beth. It's an invitation. So I'm going to tell you a little bit about what we've been working on. Some of it is basic research and how that slowly transitions to translational research because we are working in a university hospital, one of the probably the biggest in Germany. I've only recently started my own group, so you will see some things that I've done before and how it's now going towards the clinical cancer evolution aspect. It centers on using information from patients and patient material like organoids to track evolution under treatment. And you'll see that it starts with adult cancer types and slowly moves to pediatric because we are located in the pediatric surgery clinic right now. So we are interested in cancer evolution. And cancer evolution itself is a topic that has only recently started to pick up pace and interest. It all starts with the basic foundations that we have learned from Darwin and Wallace in the 1800s. A few hundred years later, Peter Noel started to describe cancer as an evolving entity, and this is the first sort of sketch that I'm in a clinic, so things ring. The first time he's had a sketch of a cancer growth. And then already in 2012, we've had the first description of an actual breast cancer evolution or phylogeny of one patient. So starting from the very beginning of the conception of a cell and how it has grown and evolved into a cancer entity. And since then, we have been able to also look at the evolution or the cells from a tumor at the single cell level. So using whole genome from single cells, we are able to extract information and watch how these single cells evolve as opposed to clusters as we've had before. And the reason we can talk about evolution when it comes to cancer is because cancer fulfills three aspects that we're interested in, or preconditions to allow you to talk about evolution. And these are that there's a variation in the population, and this is cancer heterogeneity. That this heterogeneity that we're looking at is inherited from a cell to the daughter cell, and that it can be selected, that this heterogeneity can change depending on environmental aspects or treatment, selective pressures that are out there. And these conditions are satisfied, so we can look at cancer evolution. And we've done this on different cancer types and at different levels because you look at evolution and what happens before evolution is the heterogeneity at different levels. So in a study where we look at pancreatic neuroendocrine tumors, we did a study on their methylation and the heterogeneity. So pancreatic neuroendocrine neoplasms, these are a very small subtype of pancreatic cancers, just 3%. And these PANNs, as the abbreviation, is then divided again into PANNETs and PANNECs, depending on their origin. And quite a problem in the pathology where I used to work before, for a pathologist to identify these tumor entities was really that the distinction's not very clear if you're looking at the histology. So the separation was not easy, and that was a problem for them, especially because the two different subtypes of this small subtype actually require different types of treatment, and they have different outcomes. So with a student of mine at the time, Tinsy Simon, we looked at the methylation profiles, and using that we were able to separate these tumor types eventually better than a pathologist could. And we could also use this kind of heterogeneity and phylogenetic relationship to identify the cells of origin, so the PANNETs, we found are closer to the endocrine and the pannix to the exocrine. Some of this has already been suspected, but we were able to confirm it in our study. In another level or study, we look at heterogeneity in early progression. So you don't have the tumor already, you haven't identified it, but it's just starting. And this is best identified in a study of colorectal cancer. So here, we look at progression of colorectal cancer, which is stage-wise. It's a very nice entity to study cancer evolution because the different stages are histologically clear to a certain aspect and have been well investigated. So a mushroom growth, a polyp, or an adenoma, as we call it, will grow into a carcinoma. It can invade the surrounding tissue, and that's at that point, this is the primary colorectal cancer, and it can move into the body and metastasize very often to the liver. And here, if you remove the liver metastasis, we also quite often get a relapse. So these are the samples we have at the clinic and that we investigate. So looking at the very early stage here, we were able to look at the switch between adenoma and a carcinoma. And this is because at the pathology, we had these precious samples, which are the actual mushroom, which you find. This is an image from a colonoscopy, and you kind of can see the mushroom coming up. And these, although they looked really intact, so they don't look like they're invading the tissue, still by law, a pathologist has to look at each sample. So you have to section them and look at them. And a pathologist has to say, okay, they are, let's say, they're not dangerous to the patient. And there is no further treatment required. And every now and then, the pathologist I was working with would find samples where actually the carcinoma, the cancer part, has already started to progress. So in one growth, you have the adenoma, which is the past, and the carcinoma, which is the present or the future, together with surrounding normal control. So the three stages, I always say they're caught like red-handed right in front of you, and you can see time, and time means evolution. And that's why we use these samples to study cancer evolution. And what we did several studies on these samples, but most interestingly is that we found that all these adenoma and carcinomas carried driver mutations already, like P53, KRAS, already in their adenoma, and they were heterogeneous and they were evolving differently, and we tried to make their phylogeny. But these main driver mutations were found at different stages, and they didn't necessarily have to be taken into the carcinoma, which is here in red. And that was a very interesting finding at the time. What we also used colorectal cancer for is to study later levels of progression. So this is a study on the primary colorectal cancer. It's a stage 2 tumor. And we were able to take the whole tumor out as it is, and we divided it into like 68 different parts, depending on whether it's in the invasive front or on the sides or not. And we sequenced each one of these subsamples at very high depth using a targeted panel that we designed to see, because we were like, wow, heterogeneity, if it's so heterogeneous before, this must be very heterogeneous. But we and others have already shown that actually at the driver level, there is no change in heterogeneity. The driver mutations are stable, but it is the copy number that changes really significantly here in the tumor. So this is intratumoral. And what you're looking at is a reconstruction of all these 68 different samples. If you put them back together and you relay them back to their location in the original tumor shape, you can find that copy number changes along the axis of the tumor. So here's like the reconstruction of the tumor, and the redness, the more red the color is, the higher the copy number change is. And you can see here the reconstruction. You're looking into the slide is where the lumen is. And then if you turn the tumor 90 degrees, you can see how that changes along the axis as well. And then if you take all the information from all the genes that we were looking at the copy number level and cluster them, we saw that they were sort of separated into two main clusters. And if we put these clusters back onto this 3D image, we found that they sort of separated the tumor into two parts, perhaps portraying how a tumor grows, but importantly, really showing us that there is significant heterogeneity at the copy number level in a tumor, and this needs to be taken into consideration when you're sampling a tumor. So taking one sample from one side doesn't really give you the full picture. And since then, most of my work and now my team looks at the late stages of cancer evolution or progression, which is when there is a metastasis and a patient is being treated heavily, and a tumor or metastasis can relapse. And our question is why? How can these cells who are being put under such strong selective pressure to die, these treatments we know are toxic, how come these cells, tumors come back? So we work in quite a multidisciplinary team on these questions with a team from Spain. Laura, she's a bioinformatician who works on cancer evolution. The clinician, Nathaniel Rustock here, he's the surgeon who gets these samples. And that's really a very important relationship we have, because that close network allows us to get the samples we want. And as many as we want, so multiple sampling, because as I told you, if you just take one sample randomly from a tumor, it's not going to tell you enough information. And to track patients over time, which is important for us in evolution. And in this project, we also worked with Eric, which is part of the BIH, or the bioinformatics facility here in Berlin. So what we did in this very long study that actually only focuses on 9 patients, but we go very deep into the situation of these 9 patients. They're colorectal cancer patients who have had liver metastases, and we have multiple samples from the tumor, multiple samples from their surrounding, and in some cases, information from the primary tumor. And they have all been treated. It's not a clinical trial, so they've been treated differently, but they've all had a 5-FU chemotherapy backbone. And between 1 and 3 years, all of our patients have relapsed in the liver, and they relapse in a different location, so we know it's a different growth, and they've been sampled again. And here we look at the DNA, at the RNA, and we created a database so people can look for their gene of interest. From the DNA, we can do alterations, look at mutations, copy number, and signatures, and then evolution. And from the RNA, we extract information from immune cell infiltrates, new antigens, and these kind of subclasses that are important for colorectal cancer. And the first thing you ask when you have such a nice matched sample cohort is, well, are the number of mutations changing after extended treatment and the relapse? And actually, we and others have now shown that there is no change in the total number of mutations. We look at selective pressure. This is a method called TNDS, to see if there is an increase in selective pressure on these samples that have now come up, or decrease, but we see no significant difference. And also, if we look at all these samples together as well, before and after treatment, and look for copy number alterations and gains and losses, we also don't find significant differences. Everything I'm showing, by the way, the green is like the first liver metastasis and purple is the relapse. It's how they're color-coded. So putting everything together, you're not going to see big differences. But when we started to open up and look at it by patient, we start to see differences in patterns. So some patients have less mutations after treatment, and some have much more. For example, like this is total number of mutations. And from here, we carry on with looking at the exact number of mutations. And just as a side note, if you're in colorectal cancer, so they are all MSS, microsatellite stable, except for one patient who had a MSI relapse. And it's just a different subtype of colorectal cancer. I'm not going to go into it because we're just going to look at the evolution now. So our focus is to make this kind of phylogeny, so a story, a life story in a way, a history of each patient. And then we follow how they've evolved after relapse. The purple branch is where the relapse has happened, and then the subclones are the ones related to whether the relapse happened, so where the relapse happened. And these are the main mutations, of course, not all of them, but they're the main drivers that are important in colorectal cancer. And what we find, although we have a limited number of patients, already we can see like 2 groups of patients emerging. This one group we call pre-existing, meaning that we know where the relapse is from. So if the relapsing clone, for example, is the blue one, we have evidence of it being present in the first liver metastasis. So here's an example where in one of the 3 samples, that blue clone is there. Just to make it easier, we color them like this. So that probably gave birth or further propagated the relapsing resistant clone. In another group, we don't find evidence of the relapsing clone. Here it's in green. That's the leading relapsing clone. We don't find it in the first liver metastasis. So it didn't come from something that was already there or ready to metastasize. It could have come from a different metastatic origin. Ironically, for one patient that we have because they were a patient from our hospital, we were able to go back and find that patient's primary tumor. And there we actually found a clear location or evidence of the relapsing clone, this green clone here. And it's the one that is able to actually give the relapse origin, although it has been resected already a year before even the first liver metastasis was present, indicating probably dormancy and resistance or other metastatic sites that we did not know about, because that's all we knew about this patient from the clinical information we could get. And then just to carry on looking at these two groups, we found that the pre-existing relapsing clone group, where you have a clone already found in the green, so the liver metastases, that is similar or the origin of the relapsing clone. These two patients who had that, they had also in that sample responsible for the relapse evidence of a DNA mutational signature, which is related to 5-FU, which this is a chemotherapy that all patients actually received. But only in these two, where there was evidence of relapsing clone, do we find that signature already present, sort of like a telltale of what had already perhaps been a signature of resistance already there. And then we also looked at immune cell infiltration in these samples, and we tried to see if there was a similar pattern, but that's not the case. So you have groups of patients who have decreased immune infiltration or no difference and increased. And this goes on into a different study, but so I just leave it here. We also looked at the neoantigens to say, well, after treatment with so much chemotherapy, does your average number of neoantigens change after relapse? But there is no significant difference. And looking also at patterns with this CMS, so this is a type of grouping, a subgrouping that is used in colorectal cancer, we also don't see this pattern come up. In one of the patients, we went a bit further and we did this whole genome single cell analysis. And this is in a collaboration also with the group from David Posada in Spain. This is Joao's work. He took one of our patients and took about 60,000 cells from both the first liver metastasis and the relapse. This is a patient who had 3 years of treatment of chemotherapy and VEGF inhibitor. And the liver metastasis cells are in purple and the relapse is in orange. And he made the phylogeny. And now we're not looking at clones. Like all the phylogenies I showed you before, you have a clone. We do declone evolution. We try to group cells together in patterns where they share mutations or copy number profiles, because that's all we can do. But when you're looking at single cell whole genome sequencing, each branch, each end of this phylogeny is a single cell. So that's very cool. It's as detailed as you can get. So what we find is that they, of course, share the most common recent ancestor, and these are the main driver mutations. Here are the big cluster of cells that belong to the first liver metastasis. And from one of these clades, the arm extends to the whole collection of cells that come from the relapse. So they've all very tightly related to one origin from the first liver metastasis. And what I thought was really interesting is, although we did whole genome sequencing, only 4 non-synonymous mutations were responsible for the whole relapse. And those 4, up until we made the study, had no real further information that we could find anywhere related to cancer survival, treatment resistance, or anything else. So it's probably something else that's driving this kind of resistance. And it's probably not so much at this late stage on the driver mutation side. And that's when we started to look at plasticity. So not DNA so much, but RNA. But before I jump in that, I also wanted to tell you that's also around the time when I was thinking it would be really interesting to stop looking at just the past. So everything I've shown you are patients that have perhaps already not maybe survived. These are very old material. They've been archived, and everything we learn, we now say, oh, okay, we found out this and this, but we could have done this, or maybe if something else had happened. So there was a lot of looking to the past, and that's something I also wanted to change. So now we make models of patients that are already in the hospital, and they are being treated as we speak. So we get their material. And these are now adult and pediatric children tumor samples that we can grow in these 3D organoid or tumoroid. I use the terms interchangeably. There's a whole discussion on it, and you can ask me if you like, but they are organoids from tumors from our patients. And we do many things. So we're building a platform on that. And one of the things we'd like to do is do co-cultures with immune cells from these patients, or what we've started with is fibroblasts. And for the clinic, we offer a drug kind of therapy testing. So your patient comes in, you grow their organoid, you can check a few treatments, and you inform the clinician of what you find in terms of response. But what we do is then watch them evolve. And most importantly, we interfere with their evolution, which is very cool. So you're no longer looking at the past of what has already happened. You're seeing it happen in culture, and you're manipulating it. And we do this for several types of cancer. Colorectal cancer was the main thing we started, and everyone who does, I think, organoids likes to work with that because they are quite easy to establish. So we have a nice collection. We are still very interested in these late stages, so metastasis and relapse. We try to treat them in treatments that are similar to what the patients have had in the clinic. And sometimes we have them matched, so the metastasis and the relapse, and that gets very interesting because then you can see how they respond to treatment differently. And we work with them in the lab. One experiment that is still ongoing, but just to give you an idea of how we use them in a plasticity. So this is now evolution, but it's RNA level. So it's more about the change in plasticity and expression. So we grow these organoids in large containers, and we treat them with treatments that are used in the clinic, either in a continuous scheme, so that's like having constant selective pressure, or in an off-on scheme, so giving them a break and washing out the chemotherapy. And we sample weekly. This is a small experiment we do for 5 weeks. We do longer experiments for further, and we try to follow how resistance comes up at this point. So these are also liver metastases, and we push them into becoming what would be like a relapse or resistance. And we look at the single cell RNA and try to use this information from their clusters to inform us about how they change over time and what this would mean. So for example, this gray cluster that comes up after continuous treatment and dives down, we ask, okay, well, what patterns or important pathways are they connected to? And that would be something about to do with maybe filament arrangement or something to do with the membrane. And we'd like to take that further and then ask, well, what happens if I try to block different important pathways that belong to these clusters that come up midway and confirm resistance? What we
I
also
have
do in other, yeah.
sorry, I have a question about that last slide. So you said that you assayed every week. I'm wondering if you have a more aggressive or less aggressive tumor type, if it would make sense to like change that window, or have you found that a week is like the best interval for testing?
Yeah, that's a good question because today we had such an issue with a neuroblastoma, which is a pediatric tumor. And there we found that the treatment we had was too aggressive. So we decide the treatment based on IC50s and different criteria. And then we come to sample on a weekly basis, but we just don't get enough to do the single cell analysis, like at all. And that would be a criteria to skip. And now we do 2-week intervals, but right now we're figuring it out as we go. So it started as
Yeah,
a week because we knew that was going to work for these tumors, but it's not necessarily the case.
yeah. So each tumor could be a little different. Oh,
Mm-hmm.
that's interesting. Thank you.
Mm-hmm. Yeah. Um, And with another experiment, we do longer. So this is 17 weeks, and you can see that the IC50 changes so that they become more resistant. And here we're interested in single cell RNA spatial presentation. So we look at how these cells are not just different at the single cell level, but how their locations or the clusters change. So at the top, you're looking at organoids that have had no treatment. After this was 8 weeks when we look at these samples, and the lower panel, they've had this is a FOLFOX. It's a cocktail of chemotherapies that are used in colorectal cancer. And you could right away appreciate that the orange and the green cluster, let's say, so the 1 and 2, are completely differently arranged. And really significantly, you have this orange cluster that's at the outside and green that kind of goes towards the inside. And one of the interesting genes that pop up are MALAT1. I think this was a long non-coding RNA, and it is in the green cluster that's in the middle. And this is relatively new, so we have to think about what that tells us and what we can do with that information. And here you can see it again, just on its own. So that's colorectal cancer, and that's our platform. We have primary metastasis. We have interesting scenarios where this one patient 17, has multiple metastases in one surgery. So from the liver, but also from the small intestines and the ovaries. And they all grow. We have them all as a line, as like organoid lines, and they all grow. They look very different. And we're looking at what that means also in terms of response to treatment. And we also have, like number 14, I pointed out before, it's metastasis, and we have its relapse, which is really hard to grow. So these are matched and running in other experiments. And then we moved towards pediatric cancers. And here at the beginning, we could only make PDXO samples. So neuroblastoma tumors, which is a pediatric cancer grown in a mouse and then made into organoids. And we've been investigating those. But actually, recently, we've started to show that we're almost ready to show that we have, or to use a panel we've made of neuroblastoma tumoroids. So these are really, really difficult to make and do not live for very long. You can see that as time goes by, as time moves forward, we sort of lose these tumors. But they come from patients in our clinic, and we're trying to build this as a platform with like clinical information we get from the oncologists and see if we can use it to help them decide, or give them an idea. Decide's a big thing, but to give them an idea of what sort of treatment might be informative. With these also the PDXOs that we're growing. So they're very different from adult cancer, very different from anything we've had before. They're very, they swim, they don't like to sit in matrigel. Some of them do, you can see here below, we've tried that. But they're clumps of cell, they don't really, so that's why the term organoid is a bit difficult to use here. They don't really show phenotypical arrangements or an organoid-like structure, let's say. They grow in clumps, but still very interesting because they just came from a patient. And what we do together with pediatric oncologists here is we try to get information from them when they've done, for example, here an open biopsy. So they get a lot of material. It's not just a needle aspiration or a needle biopsy, and we can grow these from the patient they're interested in. We ask what kind of treatment. So this is a treatment cocktail for pediatric neuroblastoma, and we try to replicate this and try to see if we get a relapse or resistance, that could be informative to the clinician. So we're building this very close collaboration, but it's still like just starting, because these organoids are very delicate. They will die quickly, and bringing this information to the clinic is still tricky because you can't say without large tests, of course, that this is going to be useful. But sometimes when together with our oncology colleagues, things for a patient are really in a bad way, it would be worth being able to test some alternative treatments, so different drugs. But what we do is we're building this project with a student of mine, Dad, who just started, and here we collaborate with other specialized people. So René is looking at light sheet microscopy, and what we do is we grow these pediatric organoids, co-culture with fibroblasts or endothelial in this project. We treat with different inhibitors. We're interested in targeted inhibitors that are being used now in the clinic. And we see how that changes over time. And while they or his group looks at live imaging with light sheet nanobodies that we make just for this kind of study. So nanobodies are very, very small antibodies, and they can penetrate the tissue. And we try to set up like a live imaging situation where you can sample over time and treatment. At the same time, we collect samples and look at the phylogeny of these different cell types. And everything we do is funneled into an evolutionary idea. The basis of what we want to do is be able to make organoids for patients when they come in. And as they are perhaps being treated or along that line, while we are also treating them with different types of with the same type of treatment, but doing evolutionary studies. So for example, we can barcode our cells, we can make them resistant or sensitive. That information is funneled back into the clinic and informs the oncologist whether to continue treatment. And we say always, if you're treating at the maximum treatment possible, you could land in a situation where you cause resistance and then you can't treat again. So we want to have this information coming in from our evolutionary organoid platform. And instead of waiting years and months, we can inform you about how these tumors will evolve into resistance. Yeah, so that's what I wanted to share with you today. Recently, we moved to the pediatric surgery group, but we were for a long time in the oncology, hematology for the pediatric cancer under the leadership of Angelika Eckert, who also just moved now, and it's Heidi Deubze. And I was working in the pathology for a long time, closely with the surgeons from adult cancer and the bioinformaticians from different institutes. This is my young team, especially Andrea Mena runs the lab and all the technical aspects. And we're located at the University Hospital and would love to hear back from you or see you if you want to visit. Thank you very much.
Thank you. That was a fantastic presentation. I really appreciated all the microscopy images that you're able to kind of see all of your like stains or floor like probes in 3D. So that's something that I think is really cool and kind of cutting edge, and probably an advantage over doing something like a mouse avatar kind of study with tumors. I guess my question is, can you speak more to kind of like the pros and cons of using kind of organoid model outside of a body versus like a model avatar?
Yeah. So yeah, when I show organoids in almost any talk, I always get the question of, yeah, but you're just looking at tumor cells, the microenvironment is so important. Of course, and when I'm in a bad mood, I say, well, it's better than cell lines. So I think it's like degradations, right? A lot of work is still being done on 2D cell lines that grow on plastic for 30, 50 years. So yeah, we're moved forward. Also, when we make these organoids, they come from that patient, right? So it's not a mouse model. It's not, we're really that close. In colorectal cancer, it's been really well documented that they can show the response you get from certain treatments with organoids is representative and can be used to compare to a patient. But of course, we are trying to do co-cultures with other cells from the microenvironment. And eventually we want those cells to be from that same patient. So we have fibroblast and the tumor, the neuroblastoma, from one patient that we try to grow together. There will always be that problem, but they grow fast, they grow from the patient, and they grow, maybe neuroblastoma not, but others, they really represent this 3D organization. So the cell-to-cell contact, you're getting a lot of information using these organoids, but they will lack a lot of information from the microenvironment.
That's great. That's a really great overview. I mean, I appreciate all the visuals and everything that you might not be able to appreciate while using like a a mouse model or something like that.
Mm.
So, and then the idea of bringing in more of the microenvironment sounds really cool. So thank you so much for that. I'm going to open up to any other questions from our participants today. I see Cheryl, you came off. Yeah.
Yeah, I had a question. I'm just wondering to what extent like how much data you've collected in terms of the patients that you've modeled and the treatments that you've done in the organoids, and what kind of correspondence you've seen so far. I don't know if you've done enough of them to have to draw conclusions, or how it's looking.
I haven't. So especially the neuroblastoma, we spent a year and a half trying to establish these organoids. They're extremely difficult. So I haven't, but we're moving patient by patient. With colorectal cancer, we have done a lot of treatment, but because we didn't have, so you need a special kind of ethics to go back and check if that patient responds the same way you are. So actually don't have that data. I depend on a huge number of like other studies who show that it is representative, but I haven't in the colorectal cancer either.
And are you in which is it going in the direction of like you'll mimic what they're doing in the clinic, or are you informing the clinic?
Right now we're going to mimic and
Yeah.
see if we get basic things like is that patient responding really well? Did our organoids respond really well? Just basic things to try to sell it to the oncologists, because there is a lot we have to do to convince them to look. But in the future, what we would like to do is inform. Evolutionarily.
Sure. Yeah, that's really cool.
Hi, this is Layla Mnayer. I missed the first part of this very interesting talk, and thank you so much for this. My question is pretty technical from someone who's done quite a bit of stem cell cultures. So how are these organoid cultures started?
So they come from a tumor from the patient, and then we do different mechanical dissociations to get the cells out of the tissue, and then different enzymatic digestion. The thing is, and then so because they're so different, like I keep repeating, the colorectal is a whole different world. You can use enzymes that are strong, and you can really dissociate into single cell, and then we let them grow in media that will propagate tumor cells as opposed to something else. In neuroblastoma, they are so delicate that we sort of like do mechanical dissociation a little bit and then kind of leave them alone and hope that the tumor cells will be the ones that overgrow the population. And they have so far, except for one case where we also had a fibroblast line.
Okay.
Is that what you're at? Yeah.
So the treatments that you're doing on those cells is a way to maintain the structure of the tumor cells, in addition to other cellular entities that exist within the tumor.
So what happens is, after a while, and these have been growing the colorectal for years with us, there's only tumor cells left. Yeah, they're the only ones who will survive. And we do sequencing, we do a lot of testing. It's a pure population. The neuroblastoma, you can have other cell types that hang out for a while. We haven't had them long enough for me to tell you 100%, but when we do sequencing, the allele frequency is so high for the drivers that we feel confident that it's really mainly a tumor population. We're not encouraging the growth of anything else yet.
So my question now is that how are they different from cell lines in this case? You do not demortalize, or so I'm kind of curious.
So with the colorectal, we grow them directly in a gel-like structure that gives them this ability to remain in a 3D, and it's really nice. Like you can see the cells on the outside grow next to each other in a very representative villi and crypt kind of pattern, and inside they're different. You get a necrotic core, which I find is really interesting as well, because that happens in a tumor. The neuroblastoma are spheroids in a way. They are clumps of cells that like to stick together, and they grow usually floating. They don't settle.
Oh, okay.
Yeah.
So it's pretty much the gel structure that you have that helps the tumor maintain the cell-to-cell and then the integration that is similar to what you have in the body. This is
Yes, in the adult cancers, yeah.
yeah.
Mhm. Yeah, yeah,
Thank you so much.
yeah. You're welcome.
So can I ask you, Soulafa, where are you located? I'm sorry, I jumped in.
Yeah. Oh, I'm located in Berlin at the University Hospital. This is the two main universities we have in Berlin combine their medical faculty in this one hospital. That it's called Charite.
Okay, very nice. So it's Germany. Okay,
Yes,
very nice.
yes.
Yeah. I'm in Connecticut, Hartford Healthcare, Hartford Hospital, US.
Yeah. Nice to meet you. Thank you.
Nice meeting you.
Do we have any other questions? Otherwise, I have a couple too. I just don't want to monopolize the time, but I guess I'll jump in. I'll ask a little bit about you. You discussed tumor heterogeneity quite a bit. Kind of with your experience using these tumoroids, I'm wondering if you could kind of explain a little bit what your opinion is about what's the best way for us to really biopsy a tumor in a clinical setting if we know that we're going to be missing quite a bit of information just by doing a quick biopsy.
Yeah. Yeah, it's a good question. In a perfect world, we'd biopsy, I don't know, 100 times everywhere, but that's not going
Right.
to happen. So I think everything over one sample is already moving forward, just to be aware that that one sample is really dangerous. So when we get a tumor, if we're able to sit there with it's called, I don't know the English word, where the tumor first goes to the pathology and they have to quickly cut it up. We're standing there with them. And then we look at the tumor and biopsy different locations, really actively look for different areas. So the invasive front, the necrotic core, and as many places as we can. So not just the number, but location is important.
Mm-hmm.
And then time. So I think when we get better like circulating tumor DNA, these kind of things that you can biopsy over time, it gives you also more and more information about what's happening. So it's multiple sampling and including the time, the changes that are happening. And that's the informative. Yeah.
The time is an interesting variable, I think, because it seems as though this technique might help inform like appropriate timing for longitudinal studies of tumors and things like that. Are you seeing that the use of organoids might help us understand better when to perform longitudinal studies on tumors?
Yes, that's the big goal of the big pot single cell plasticity studies I'm showing you with organoids, because when I first started going to the surgeons to ask for samples, I kept wanting to get more biopsies, and they're like, you can't biopsy a liver metastasis in a really unwell older person. That's not ethical. And so time becomes hard to grasp. And with these organoids, we sort of create that time for us. You can sample as much as you want and you can do as many studies as you want. And eventually, I'm hoping we can see these bottlenecks in the evolutionary context and then know that this is when it would be smart to change treatment or intervene with the patient.
Mm-hmm.
Yeah.
Well, that's really
My
cool.
problem with the circulating tumor DNA is a great field, and we have a lot of people here working on it. The problem is for an evolutionary study, you can't always be sure if you don't find a mutation in circulating tumor DNA that it's not there because it's sparse. So, and that's a problem when you do evolution. If something's not there, is it gone? Did you lose the clone or did you just not sample it? It becomes more and more difficult. Yeah.
Right. And then in addition, the circulating tumor DNA, if you have multiple tumors, it doesn't tell you where it's from either. So I feel like it's an imperfect, there's no perfect technique, right? So it's just understanding what the advantages and disadvantages are of each one.
Yeah.
But super cool. Thank you.
there's work in methylation with circulating tumor DNA, and that can perhaps inform of the location of the metastasis.
Ooh, that's
Yeah,
cool.
it's coming up, I think. Yeah.
That might be interesting for like the carcinoma of unknown primaries and things like that. Is there an
Absolutely.
application for organoid use for carcinoma of unknown primaries, like seeing patterns in the different, I guess, staining and markers and things with
Yeah.
organoids versus other techniques?
Yeah, I actually don't know, but
Mhm.
when we did that methylation study with the pancreatic cancer, that was really on my mind, like carcinomas with unknown origin, because we were able to track the origin of these two different subtypes of the pancreatic neuroendocrine cancers so well. And I was thinking this must be done, and it is being done everywhere. Like many people are looking at methylation patterns to identify it. But with organoids, it's very interesting. I actually don't know what is being done.
Mhm.
That's an interesting project.
Yeah.
Because at the hospital, you can get these samples. That's
Mhm.
possible.
Interesting. Oh my gosh, I'm really looking forward to kind of following this work now. Thank you so much. I had one more question, but anybody else with a question, please interrupt me. The last one I have here is, do you find differences between a relapse at the primary location versus a relapse at a metastatic location? And kind of what differences have you found with that?
Yeah. So we don't look at that, and we really actively use different words. I think that relapse at a primary is more like a recurrence. It's just because we need to use just a word to describe the difference. And relapse in a metastasis location is what we call the relapse here. So we did not look at the primary recurrent, or yeah, relapse in the primary. I don't know how that would
Mhm.
look like. We're
Huh.
really actively looking at the late stage.
Late stage, like
Distant
relapse at
distant
the
location.
Met. Okay,
Yeah,
cool.
yeah, yeah.
All right. Well, that's all I really have. Um, does anybody else have any questions from the participants today? Twice. Well, thank you so much again. This was like really insightful and really helpful. So thank you
Thank you
so
for
much
having me.
yeah, thanks for coming in later hours. I know it's close to 6 o'clock in the evening where you are right now,
That's
so really
fine.
appreciate it. And
My
thanks for all
pleasure.
the work you're doing. This is really interesting, something I'm going to have to follow up with in the future. So what interesting and great work, and imagery, like the microscopy is really something. So thank you so much.
Thank you for having me. Lovely. Thanks.
Thank you.
All right, everybody have a
Thanks.
great week. Well, we'll see you in 2 weeks. Okay, bye-bye.